Hi, my name is Onyekachi Ibe. Today is the 2nd of May 2025. So, today I'll be presenting on one of the most fascinating sensory processes in the human body. That is how sound waves, just pressure fluctuations in air, are transformed into electrical impulses that our brains interpret as meaningful sound. This process called transduction is at the heart of auditory perception and is a beautiful example of coordination and communication within the nervous system. What is transduction? Think about this. The reason you're hearing my voice right now is because a sound wave physically moved a microscopic structure in your inner ear, triggering an electrical signal. That one moment of movement set off a cascade of communication that ended with your brain decoding my words. Transduction isn't just a concept, it's the reason we can speak, listen, and interact with the world. How sound waves travel, the mechanical wave pathway. The auditory journey begins in the external ear, where sound waves enter the canal and vibrate the tympanic membrane or eardrum. These vibrations are then passed to the ossicles, where tiny bones in the middle ear that amplify the mechanical signal. The final bone, the stapes, pushes against the oval window of the cochlea, converting the vibration into a fluid wave. This marks the transition from air-based conduction to liquid-based conduction, a key step in auditory processing. Inside the cochlea lies the organ of corti, the actual site of sensory transduction. The basilar membrane vibrates in response to the fluid wave, and this movement pushes the hair cells, that's the sensory receptors, upwards against the tectorial membrane. The hair-like projections of these cells, called stereocilia, bend in response to this notion. Each step in this process is coordinated so that mechanical energy is directed precisely towards electrical conversion. When the stereocilia bend, tension is applied to tiny structures called tip links, which open potassium channels on the membrane. Uniquely, the surrounding endolymph is rich in potassium, so potassium ions rush into the cell, causing depolarization. This electrical change opens calcium channels, prompting the hair cell to release the neurotransmitter glutamate. That glutamate then stimulates the auditory nerve, formally converting mechanical information into neural code. Hair cells communicate using graded potentials, small voltage changes that vary in strength, but neurons operate using action potentials, that is, all or nothing spikes. The hair cell's signal triggers this transformation, allowing precise transmission of sound data to the brainstem. Importantly, the cochlea is organized tonotopically, meaning different regions respond to different sound frequencies. This spatial encoding is preserved all the way up to the brain, allowing us to distinguish pitch and tone with remarkable precision. Unfortunately, this system is delicate. The hair cells can be damaged by aging, loud noise, and certain medications. Since they don't regenerate in humans, damage results in permanent sensorineural hearing loss. In some cases, cochlear implants are used to bypass damaged cells, entirely directly stimulating the auditory nerve. Genetic mutations affecting tip link proteins can also cause congenital deafness. These clinical examples underscore the importance of proper transduction in maintaining sensory communication. Transduction is where physics and physiology meet, transforming a sound wave into a meaningful signal the brain can interpret. We've explored the structures involved, the ionic mechanics, and the clinical consequences when things go wrong. This process beautifully illustrates coordination within the sensory system, from hair cells to neurons to cortical awareness. To help solidify your understanding, I've created a worksheet to trace the full sound pathway. Thank you for listening.